Okay, so this is just a still shot that I grabbed from a Wikipedia site that helps you think about diffusion a little bit better. And if you see, this is just like a slice through a beaker or a tank, and this dashed line in the middle represents a semi-permeable membrane. And what that means is that there are gaps in the connection between here. So it's not a solid wall. And the semi-permeable means that some things can pass through and other things can't. And the thing that prevents them from passing through is their size, not their charge. So the semi-permeable membrane in the lab setup that you would be doing this week or in any other scenario basically represents the cell membrane, because the cell membrane lets some things pass through it and prevents other things from going through, both due to size and charge. But in this example, we just are considering size. So these little red dots are small enough that they can actually pass through the gaps in the membrane. And that means that if we start out with a really high concentration here on the left and a low concentration on the right, over time, we will end up with equal concentrations of those solutes, those red dots, on either side of the semi-permeable membrane. And in fact, you can imagine that those solutes, those dissolved particles, would be able to pass back and forth across the membrane. But once equilibrium has been reached, we're going to maintain a balance here. And unfortunately, I can't really draw big objects, but if you pictured something like, connect the dots between 4 of these guys and think of a cube, and you could imagine that that was then too big to pass through this opening. And so even if we had a whole bunch of those cubes here on the left side and only 1 or 2 over here, the membrane would not be permeable to those larger objects because they would be restricted due to size. So diffusion is essentially when a solute, a dissolved substance, moves down its concentration gradient from high concentration to low concentration. And if it's going to cross a membrane or the cell membrane, then it has to be able to fit through. So diffusion happens down a concentration gradient, and it's based on permeability. So when we look at the cell and we think about what's happening in the cell, things can go through the membrane without the help of a protein down their concentration gradient via simple diffusion. So now I'm going to pull that image up that we had last week and just talk about it again. And I'll put it in slideshow mode so that you can see it really big, okay? So here on the far left, A, where it says simple diffusion through the phospholipid bilayer, is essentially what I was just showing you in that other diagram, but actually with a model of the cellular membrane. And so this is the phospholipid bilayer, and oxygen is small enough and it's nonpolar, and so it can slide right between the phospholipids because they're not actually bonded together. So small uncharged particles are the easiest to get through the plasma membrane. Small, slightly polar molecules can actually pass through, but their rate of moving is slower. And so we see in this setup, we see them moving down a concentration gradient because there's more on this side than on that side. If it were the other way around, where there was more inside the cell and less outside, then diffusion would occur down the concentration gradient out to the outside of the cell. In B and C, as I said on Monday, these are facilitated diffusion, where there's a protein that's embedded all the way through the phospholipid bilayer. So it would be an integral protein. And in B and C, what we see are channel proteins, where they have an opening that's created by the three-dimensional shape of the protein, the way those amino acids are sequenced and the way then the chain is folded back on itself. It actually creates a tube or a tunnel through which specific molecules can travel. And it's possible for molecules to be too big to pass through a channel protein. They are specific to molecules so that the cell can regulate what goes in and out. So then we have here, the example is chloride ions, which are charged, and so they don't readily pass through the phospholipid bilayer on their own, but they can move through a channel protein down their concentration gradient from high concentration to low concentration. And if we think about nerve cells, they actually have channel proteins that are kept closed, and the way that the cell creates an electrical charge to send a message is by quickly changing the internal and the external charge down its long membrane between the inside and the outside. And so it's really cool that neurons can use polarization or creating concentration and ion gradients on the inside and outside based on both facilitated diffusion and by pumping ions in the wrong direction down, up or in the reverse of their gradients. So then the third scenario is still facilitated diffusion. So it's down a concentration gradient, but this time it's water that's moving. And so water would be moving from a place where there's not a lot of solutes interacting with the water molecules to a place where there is a lot of solute. And so water moves effectively down its concentration gradient from high free water availability to low free water availability. But it's also accurate to say that water follows solute. And so the water will flow through its specific channel protein from a region where there's less solute interacting with the water to high solute. And then finally, the last scenario is with facilitated diffusion, where you see that we're still moving from high concentration to low concentration. So down the concentration gradient. So it's still considered diffusion, but the protein actually changes shape. So instead of calling it a channel protein, we call it a carrier protein. And none of these requires the use of energy, which is predominantly through ATP in cellular work, because it's all down a concentration gradient. It's all considered passive. And so these are the types of passive transport. And then a couple of slides later, I have active transport. Bear with me here. Okay, so there are several slides that explain active transport. But what I wanted to show you was this diagram where you see another type of protein embedded through the phospholipid bilayer, so another integral protein. And you see that there's a spot in the protein for ATP, an energy carrying molecule, to come and bind. And when it attaches, it actually changes the shape of the protein so that a molecule can be moved against its concentration gradient, so from low to high concentration. And in that way, the cell is able to pump things against their concentration gradient and then create a really high concentration on one side of the membrane and a really low concentration in the other. And like I mentioned with the neurons, and you'll also see this when we talk about the mitochondria in chapter 4 next week, that the cells use that gradient, the high concentration on one side versus the other, to then move the molecules back through different proteins and create more ATP, which is used to do cellular work. And so active transport, the key to remember there is it's moving molecules against their concentration gradient or from low to high solute concentration. And another way that energy might be needed in the cell having to do with the membrane is when we're talking about the cell membrane actually changing shape and pulling in objects from the outside. Penocytosis is where the cell membrane pinches off little spheres or vesicles and captures fluid from outside the cell and brings it in. Receptor-mediated endocytosis is when there are specific molecules that bind outside the cell that triggers the pinching off of vesicles to be pulled into the cell. And then phagocytosis is when a cell wants to capture really, really large particles and engulf them. Okay, so this slide shows you pinocytosis, and the yellow line at the top is the cell membrane. And so 1, 2, and 3 show like a slice side view of the plasma membrane pinching off and then pulling in just liquid from the extracellular fluid. And the diagram, oh, I'm sorry, I didn't realize I did that with my mouse. So down below, you can see 1, 2, and 3 again, but it's an actual image taken with an electron microscope that shows the little vesicles forming and pinching off to pull fluid into the cell. So, you know, how does a cell get more liquid inside? Well, it's through pinocytosis, which is cell drinking. Okay. This next one, the yellow is still the cell membrane, but you see the purple along the edge on the interior of the cell is a protein that is lining the inside of the membrane of that phospholipid bilayer, and then going through it would be some other proteins and then attached to these receptor molecules that are hanging out there that would bind specifically. And when things bind, they trigger a reaction, and you can see through steps 2, 3, and 4 that it causes the cell membrane to pinch off and bring those specific molecules in. And that would be if the cell wants to bring things like glucose in and it wanted to bring it in quickly. Usually there are channel proteins specifically for glucose, but there are molecules that are bigger than glucose that wouldn't really fit through a channel protein, and so the cell would use receptor molecules for those. And then down below, steps 1, 2, 3, and 4 actually show you that vesicle, but you know, with a slice through it, so you don't see the whole sphere being brought into the cell. And then this last one is probably familiar to you, where a cell might engulf an entire another object. Maybe it's a microscopic particle, maybe it's an actual other organism. Part of our immune response is actually that our white blood cells that travel in our bloodstream, they attack foreign objects, things that are in the blood, like bacteria, and they engulf them through phagocytosis, and they make a vesicle and they trap the whole thing inside, and then they use lysosomes to merge with that and, you know, basically digest or break down the foreign body while protected inside the white blood cell. I'm going to pull it into view all of the slides. Oh, I didn't talk about exocytosis. So endocytosis is bringing things in. Exocytosis is where we are having things leave. And so the principle is similar, but at this time, instead of bringing something in, we're taking a vesicle and we're fusing it with the cell membrane, and then we're releasing the contents to the outside. Anytime somebody scares you half to death and you have an endorphin rush, or, you know, other hormones where you're scared, that's because some of your endocrine cells just released a bunch of hormone into your bloodstream through exocytosis. My sister and I used to joke we could write a book, 101 Ways to Freak Yourself Out. So she'd laugh if I remember about me remembering this with exocytosis. The lab, let's see. There are some really good videos as far as the lab goes, and I just wanted to show you on the homework that I was talking about the other day, and I found a link that wasn't working. Just gonna pull it down here and open this up. I made an adjustment, and there are 3 videos in here that really help explain tonicity and osmosis and what happens to red blood cells, and also what happens to plants. So let's see if I want to talk about this a little bit more today. Maybe
I
I'll
did. I
Yeah.
did have a question
Go ahead.
about learning assignment number 10. It seemed to make sense while you were explaining it on Monday,
Uh-huh.
but then I got to the first question and it said, identify at least 2 organic functional groups in a phospholipid molecule. And I fell
well,
apart.
we walked through that. It's at the very beginning of the video.
Oh, good. Then I'll rewatch
Yeah.
it. I think I joined a few minutes late. Okay,
Yeah.
good.
Yeah, it's at the very beginning of the video, and I pulled up the PowerPoint from chapter 2. And
Okay.
really, functional groups, phosphate is a functional group, CH3 is a functional group. It's just recognizing that there's more going on here than the hydrocarbon chains, those 2 fatty acid tails. So just thinking about, hey, this is polar because it's got a phosphate and it's got these other things that are attached here.
Okay.
So yeah, we walked through most of the questions that were in learning assignment number 10. But the videos that I'm talking about are relevant, and they're in here. Oh, I may not have updated them in the actual file, but I put them in the directions, and I've got them updated in my file now. But those are the ones that would help you fill in hypotonic, isotonic, and hypertonic. And it might help if I just pull up. I'm just going to pull up our original lab, the lab that we would have done if we were on campus for osmosis, because it has a couple of images, I think. If it doesn't, I'll pull up something else. Oh, is this the one where we have to, no, it doesn't, because we draw them. I'd spend a bunch of time drawing them on the board. So here, let's do. Normally we do 6 labs all in one big test. So this is the test actually. And I'm just gonna put it as 1 page right now. And talk about tonicity and isotonic, hypertonic, hypotonic here while these 2 things are up. And so in these 2 scenarios, we've got a sample cell in here in this bag made from dialysis tubing, and it's in a beaker of liquid, okay? And so there's a scenario, beaker 1 and beaker 2. Now beaker 1 is 25% sucrose inside the cell. So it's sucrose and water, and then it's in a beaker that's just plain distilled water, okay? And on the right side, beaker 2 is 25% sucrose, so 1 part sucrose to 3 parts water, and then 5% sucrose, which would be 1 part sucrose to 19 parts water. So it's more concentrated here than it is out there, but it's still sucrose solution in sucrose solution. So when we look at beaker 1 and we want to talk about its conditions, we would say that this bag, because it's a sample cell, when we talk about it, we're going to use the term osmotic, not tonic, okay? Now, there's a difference between the inside and the outside, so we're not going to use iso, which means the same. And because 25 is greater than 0, we would say that this model cell is hyperosmotic to the hypotonic solution, okay? So I'll type that out here. So we would say that we have a hyperosmotic cell in a hypotonic solution, okay? Now, if sucrose can't move across that membrane, which way will the water move? So the water moves into the cell, and that's because water follows solute, okay? And what will happen to the weight of that model cell? The weight of our cell will increase, okay? Now over here, when we talk about beaker 2, we are still looking at 25 being greater than 5. And so we still have a hyperosmotic cell in a hypotonic solution. And which way will the water move? Well, there's more free water in the beaker in the 5% solution than there is inside the bag, because there are more molecules of water being occupied dissolving the sucrose inside the bag, and so the water is going to flow in. And so we still say we have water moves into the cell because the water follows solute. And looking at it, we still say that the weight of the cell will increase, okay? But of these two, if they're both left for the same amount of time, in which one will the weight of the cell increase be greater? Well, the concentration difference is greater in beaker 1 than in beaker 2. So at the end of the time, so if we had, time for 60 minutes, then the weight in beaker 1, and I should be careful to type this out here, the weight of the cell in beaker 1 will increase more quickly than the cell in beaker 2. And why is that? Because the concentration gradient is higher. So it's a greater difference between the inside and the outside. And in the lab, we call that flux, or there's an equation where you can measure the rate of diffusion, and it's based on two things. And so I'm going to pull up, because I have it up right now. There's this equation here. And I'm just looking for it in the lab. That's all the setup.
And you're looking at the lab that you would normally do in person?
Yeah, I just knew that there were some images, but I can also pull up, I'm trying to remember if I saved it. I can pull up the other lab. I just need to grab Canvas here.
That would be helpful.
Yeah, I couldn't remember if I had saved it or just if I threw it in here. Okay, so if I open the lab, and I'll make sure when I exit that I save it, and now maybe I'll just have it twice. Just making it a little bit bigger. So the other professor who put this together did an experiment and created some demos, and basically you're looking at at different sugar solutions and how water moves and how sucrose and glucose move, and it's going to depend on what that membrane of the sample cell is permeable to, and whether it's, and so glucose might move but sucrose might not, depending on what the membrane is permeable to. Okay.
Well, it was actually that exact spot right there. I've been working on the lab for a few hours, and it was number 4 right there, where I thought I was understanding things, and then all of a sudden I didn't.
Okay, and I'm looking.
4 and then number 5.
I'll come back because I'm looking here, and the flux equation should be here. Here we go. It's right here. Okay. So flux is just a term for how do things move, how do they flow? And it's based on two things, P being the permeability of the membrane, and delta C being the concentration difference between one side of the membrane and the other. So if P is 0 for a given substance, if the gaps, the tiny gaps in the membrane are too small for the object to pass through, then permeability is 0. So it won't matter if there's a concentration difference on one side versus the other. If it's 100 molecules on one side and 0 on the other, if the membrane's not permeable to that substance, then P is 0, and there will be no flux. There will be no flow of solute down its concentration gradient. If I have all my doors and windows closed, a fly can't get in. All right, sorry, my internet kicked us all off. It was bound to happen eventually, but we made it like 3 weeks without it, right? Okay, so let me share my screen again. And I'm gonna reopen the chat box. And then guess I get to have a split recording. Oh, I guess it's recording. But what I'm actually going to do